Good morning to everybody. My name is Simone Barbato. I am the Technical and Sales Manager of the AgroVet division of SILAGE and SACCO Group. I've got a bachelor degree in agricultural sciences with a master's degree in food industry and enology. After some practical experiences in the wine sector as a winemaker in cellars of Piedmont and Tuscany, I worked for 3 years as a regulatory affair in the Italian Federation of the Chemical Industry. Because of my great interest in the commercial aspect and the biotechnology sector, in my early 30s, I chose to move to SILAGE SACCO Group, starting a new adventure in the agro-zootechnical division. Just a short introduction about my company. SACCO System is the combination of international biotech excellence applied to the food, nutraceutical, and pharmaceutical industries. SACCO System slogans supporting food culture and life communicates the intent that our company's mission is to support the culture of food and health, but also to develop culture, as for instance bacteria, for the food and the health sector, applicable to both humans and agricultural animals. System expresses the extraordinary synergy between 4 companies, Calificio Clerici, SACCO, Centro Sperimentale del Latte, CSL, and Chemicalia. It is a unique and functional network that maintains the characteristics and productive specificities of each entity. SACCO is an Italian family-owned company that offers a large range of innovative products. This includes starter culture for food fermentation, in particular dairy, and nutritional supplements, probiotic culture, as well as instruments for the improvement of food. The sister company, Calificio Clerici, has been an Italian leader in blended production since 1872. And in 2006 acquired the Swedish company Chemicalia. SACCO furthermore acquired the Italian cultural producer CSL in 2015. SACCO System has 7 production plants set in more than 110 countries through a network of agents and distributors. And in 2018, it generated a total turnover of 108 million euros. There are more than 50 researches and development collaborations with universities and technical centers all around the world. And our collaborators come from more than 20 different countries. This has created a unique intellectual and culturally diverse platform to grow from. Our business units are culture and probiotics, enzymes, as for end production, additives and coatings, and the labware division. Let's get down to the topics of today. We are going to discuss about the hidden world of silage, focusing on the 3 most important aspects of the silage fermentation. What is the fermentation to seek? The evaluation of the obtained final silage, and the choice of a proper starter culture. Now we can move to today's topic, the silage. As CSL, Centro Sperimentale del Latte, and SACCO, we are a producer of technological additives for the production of silage. So we can start by defining the target of our lactic acid bacteria, the silage. Producing a good silage is the main target for any farmer. Silage is the result of the work of a long year, from the planting of the forage crop through the ensiling operation until the final opening of the silo. The farmer spends a lot of effort in terms of time and money, and it's necessary to meet all the requirements. What is the main target? Production of a high quality, highly nutrient food for the animal, reducing the losses of dry matter, the biological value of the silage. What is the silage? Silage is an acid food preserve with a pH at least below 4.5. Remember that the pH is related to acidity. To improve the acidity, you should reduce the pH. How we can obtain an acidification, a faster acidification? It's possible to obtain through the addition of chemical acids, such as propionic acids, or through natural acids produced by lactic acid bacteria during lactic fermentation, as for the natural cabbage preserve that we are seeing in the picture. This level is able to deactivate the enzymes, which may decrease the nutrients in the forage, and to avoid the activity of pathogens as Escherichia coli and Salmonella. Production of a good salad with a higher protein content allows reducing the use of expensive concentrated feed as a nutritional source in the diet of cattle. As we will see, faster is the acidification of your forage, and higher will be its nutritional value. More powerful will be the lactic acid bacteria used for the fermentation, and better will be the final result. Today there are a lot of different kinds of silage, each one with specific nutritional characteristics. There are leguminous plants, the highest for the protein intake, grass or gramineous, or other mixture with the autumn-winter cereals and legumes, or the maize, the standard maize. This last one can be full body ensiled, or you can just ensile some specific parts such as the pedicle or just only grains, the so-called mesh. Obviously, the nutritional value of the forage will be different, varying the ratio between protein, sugar, and fiber content. What will never change is your target, obtaining the highest quality of silage. How can we do that? Working as best as we can throughout this cycle. So, start harvesting a high-quality forage, improving the ensiling operation, and obviously guide the fermentation process in the only direction that you need, that you want, the acid fermentation. For this purpose, nothing is more successful than a homofermenter Lactobacillus plantarum bacteria. When you have got only one correct direction to take, to choose the way to go is very easy. Unfortunately, that doesn't mean that it will be an easy journey. All along the road, there are different obstacles, such as the quality of the crops, the correct management of the silo trench during preparation, and a proper selection of the microorganisms involved in the process. You can do that by reducing the chance of contaminating the product, applying the best hygienical practices, and selecting the best lactic acid bacteria. Without these steps, the fermentation will go out of hand because of the activity of Clostridia bacteria or yeast, which would cause the loss of the nutritional value of silage and increase of the pH and temperature. Summarizing, the most important rules are three. First of all, harvest the crop at the right time, finding a compromise between the nutrients values and the low humidity. Higher is the humidity and the higher is the level of activity of all the unwanted microorganisms. The air is another important factor affecting the yeast activities, their reproduction, and the alcoholic fermentation. So a good compression of a forage crop inside the trench is needed. Even if you pay attention to any single step, the environment is very rich in natural epiphytic microorganisms. So it's necessary to use a starter culture with a high concentration. The selected bacteria must be 10 times higher than the concentration of all the other microorganisms. So there is another interesting word needed in this process, the rapidity. With the shredding, all the sugars of the forage cells are exposed to air and available for the fermentation of other microorganisms, as for instance the coliforms and the yeast. Their goal is not the same as ours. They don't want to produce a good feed for the animal, but they want to survive, eating our nutrients. So what we have to do is reducing this exposure time. During this period, we observe the presence of air, activity of microorganisms, and increase in temperature, sometimes still 32 degrees. Adding to the harvested crop a highly concentrated starter culture with a dominant strain of lactic acid bacteria is very important. If we work fine after 2 days by the preparation of the silo trench, we will observe the two most important clues in order of importance. A pH below 4.2 with the safety level of acidity for inactivation of enzymes and non-action by the pathogens, and a temperature below 32 degrees. If we meet these two values, these two targets, we are in the right direction. A safe and palatable silage with high nutritional values. In contrast, a higher level of pH and temperature are indicators of the other unwanted fermentation, the butyric and alcoholic fermentation. The result will be a toxic silage with a low amount of proteins. So, unfortunately, there is not enough awareness about how much important is the quality of the silage. If given to the animal, a low-quality feed will cost the farmer a high price, the intoxication of the animal, the veterinary costs, and the loss of quality of the production. If we will have a high level of spores produced by Clostridium thyroputiricum, this microorganism is highly resistant to the low pH. That's the reason why we need a quick and acid fermentation, avoiding its activity and production of spores. Its spores are very resistant, and they can arrive at cheese, causing the late swelling effects. How can you avoid this situation? Maintaining a right hygienical conditions, both in the field and in the trench. Avoiding to choose a ground level clear cut during the harvest. I fully understand that in this way you can obtain a lower yield, but you reduce the unwanted microorganism and so the competition for the starter culture. Choosing a starter culture with a high amount of bacteria, at least 1 million of cells per gram of silage. On this aspect, you have gotten the full control of the season. Once you loaded the trench with the forage, remember to compress it with the wheels of the tractor. In this way, you will remove the air from the silo. Finally, complete your work with a suitable covering of the silo with the right high-quality plastic material fields. Probably you have already understood how important it is to begin the fermentation as soon as possible. And to do that, you need a strong lactic acid bacteria with a high dose, because it's the only way to take control of the fermentation, avoiding the reproduction of butyric acid bacteria and their spores. The problem is that even if you have done a great job, you will ever have a certain level of contamination by the coliform and Clostridium bacteria. The ground and the manure may accidentally enter in the silo, inoculating a high amount of yeast and non-lactic acid bacteria. And we are talking about a very high amount of bacteria, the order of 10 to 5 cells of coliformity unit only in ground. If we don't use a starter culture with a higher load, the selected bacteria will be overcame. Definitely, we can summarize a certain number of best practices for producing a good silage. Choose the correct time for the harvest of your forage crops. The agronomist may help you, and in difficult situations with low sugar or high humidity, a good pre-wheating or sugaring may be a good solution. Reduce the level of contaminants with a proper cleaning and sanitization of trench. If the walls of your trench are damaged and their surface rough, it's better to cover them with a good plastic film. Preserve the hygiene of the crops, choosing a higher cut, avoiding incorporation of ground, and maintaining clean the wheels of the tractor before the compression. Maximize the extraction of the sugar with more nutrients for the good selected lactic acid bacteria. With a high dosage, at least 1 million of coliformity unit per gram of silage. Apply a good compression to the shredded forage, removing all the oxygen by the mass. And then finally, cover the silo, avoiding the excess of air and water. On the market, there are good solutions as white plastic material, which may reduce the entering of oxygen. According to the scientific studies, that's the best way to protect the silage by oxygen. One of the most difficulties in judging the silage is that we don't eat them directly as humans, so we can't directly appreciate what are the differences in taste and smell. Even if the cows cannot speak with us, we can use different instruments to obtain objective results. Most of the time, the farmers cannot see the difference. Still, there are two different ways to observe the traceable changes. Chemical parameters, as for instance the pH, and sensorial evaluation with visual and olfactory markers. The pH meter is very simple to use and offers a quick feedback about the outcome of the fermentation. As a SACO group, we have got a dedicated laboratory division with a full offer of instruments. During a fermentation process, the silage undergoes a large number of chemical reactions that can be evaluated through certain analytical determination. Having the opportunity, we suggest building a complete data set. The most important information are related to the nutritional composition of your forage. Through the three parameters of the neutral detergent fiber, acid detergent fiber, and finally the acid detergent lignin, we can build a fiber profile with a distinction between the cell walls components and the nutritional composition of your forage, and the lignin, the cellulose, and the hemicellulose. Through these three parameters, together with the soluble sugar and the dry matter, we evaluate not only the digestibility of the fiber, but even the overall nutritional values of the feed ratio. Other parameters important, as buffering power and ammonia, are related to the level and the quality of the proteins. Forage, as alfalfa and other leguminous plants, have got a higher buffering power because of their higher proteins level. Because of this richness, these forages require a significant attention for the acidification goal. The high protein content is against the acidification process. So the concentration of ammonia at the end of the fermentation offers instead an indicator of the activity of unwanted bacteria. The coliform and Clostridia damage the structure of the proteins, producing a higher amount of ammonia. So what are the most important parameters that you have to take in consideration for evaluating the quality of your silage? First of all, the pH, because it's the most immediate indicator for an excellent acidic fermentation. As I told you, after 2 days, we expect to find the values below 4.5. Remember that higher is the acidity and lower is the pH. The ammonia is related to the loss of the proteins, as I already told you, and the activities of Clostridium and coliform bacteria. The butyric acid is the worst indicator because it's an indicator of butyric acid bacteria fermentation and production of dangerous spores. A low pH is needed to stop these bacteria. Especially the Clostridium thyrobutylicum is the most resistant, as it may survive until a pH of 4.2. That's why you need a fast acidification within the first days. You can do it only with a high charge of selected lactic acid bacteria. The optimum of these parameters are forage dependent. Higher will be the protein and humidity content, and higher will be the acceptable values. That's why the leguminous plants and grass silage with a dry matter between 25 and 35% of dry matter have got higher values of tolerance for pH, ammonia, and butyric acid. So the temperature is the physical parameter of the fermentation process. It's important, but it requires a good interpretation. Many people think that once opening the silo, the only low temperature is a quality indicator. It's wrong. The temperature at the opening of the silo is related to the temperature when it was prepared. That's the reason why you need to measure the temperature on day 1. An increase in temperature during the first day of fermentation is normal. It depends on how much bigger is this increase. The temperature between 30 and 40 degrees is okay, but when you measure a temperature higher over 40 degrees, you are sure that a alcoholic fermentation is happening. There are 3 main factors which affect the rate of the cooling. First of all, the mass and the humidity of the silage, the external temperature, and the activity of the yeast. So, how to control the temperature? That's one of the most frequent questions, but we have got the answers. First of all, the temperature and its maintenance are strictly related to the stability of your silage. We can distinguish between 2 periods of stability. The first one during the fermentation process, the so-called anaerobic stability. And a second one, anaerobic stability, related to the storage of the silage after the opening of the silage. In every period, the worst enemy is ever the same, the oxygen. If you avoid the exposition of your silage to the oxygen, you have been winning the battle, saving all the nutritional value of your silage. The stability during the fermentation can be obtained only with a faster start of fermentation that creates an anaerobic environment, without oxygen. You can do that only with a powerful lactic acid bacteria with a high dosage and a great acidification power. Don't forget what is the real goal of the fermentation: obtaining the best silage with the best fermentation results. High dry matter and low pH. Homofermentative bacteria are the best in terms of fermentation results. The stability of the silage after the opening of the silo can be obtained, minimizing the exposition of the front to the oxygen. How? Through a good management of the unloading operation. When you need to cut a new slices of silage, you have to do it evenly all along the front. In winter, remove a slice of 15 centimeters of silage every 4 or 5 days, while in summer, with the higher temperature, remove at least 25 centimeters of silage every 2 or 3 days. After the removal of the slices, remember to cover the front of the silage with a high-quality plastic material, minimizing the exposition to the oxygen. If you don't cover the front of the silage, the temperature is doomed to increase. The suggestions that I give you right now are the most efficient against the activity of yeast and against the increase of temperature with a loss of dry matter. Anyway, there is another kind of useful assessment that can make our goal easier: the sensorial evaluation. Our eyes and nose cannot be as effective as mechanical analysis, but are quicker and sometimes very useful. The experience makes this method more specific and useful. The brightness of the silage is the essential quality that the silage should have. Colors as green and green yellowish, according to the species of the specific forage, are an indicator of a good fermentation and a high quality of your forage. The other way around, matte colors as green, grayish, brown, or sometimes even black, are related to putrefactive activities of Clostridium and Maillard reaction. If you never heard of Maillard, it's the same chemical reaction that makes brown the bread crust. It is due to the high temperature related to alcoholic fermentation and so to the activity of the yeast. Monascus purpureus is less frequent, but can lead to the development of a characteristic pink mold all around the silage surface. In relation to the smell, the evaluation is even more comfortable because the only good indicator is a slight acetic acid smell. Lactic acid, the most abundant acid, has no smell at all. All the other chants, starting from the putrefaction, rancid butter, the fruits, and even smoke, are symptoms of unwanted fermentation. Putrefaction is related to the Clostridia, rancid butter to butyric acid bacteria, and fruits to yeast. So the burnt smoke flavor to the Maillard reaction. So every time I spoke about the other microorganisms than Lactobacillus plantarum, I never say good things. The reason is very simple. With the production of silage, as for any different situation, you have to define an expectation and then make the possible to delete all the barriers on our road. In our case, the barriers are all the other microorganisms. Non-lactic acid bacteria, as butyric acid bacteria and their spores, are very numerous in the ground, in manure, animal carcasses, sludge water, and vegetable resides in silage trench. Even if you work properly, some of them would be present in silage. The epiphytic flora naturally present in the silage contains obligatory heterofermentative bacteria, which are producers of carbon dioxide and lower amount of lactic acid. If you want to obtain silage with the highest amount of dry matter and nutritional value, you should reduce the loss of nutrients without any production of carbon dioxide. What you need is a homofermentative bacteria producer of lactic acid. Higher will be this acid content, and higher and quicker will be the acidification. So Lactobacillus plantarum is the most suitable bacteria on this kind of battlefield. CSL and Sacco Group work in the milk sector by more than a century, leading all the evolution of this industry. The knowledge of all the technological processes involved in the industry make easier the research of the most suitable bacteria as a technological edit. Lactobacillus plantarum is the best producer of lactic acid and at the same time may produce some acetic acid and other metabolites that can inhibit the yeast and the mold development. So Lactobacillus plantarum is the bacteria that best meets your needs. A fast decrease of pH to the safety level and a best utilization of sugar for acidification purposes without any loss of carbon dioxide. Is the lactic acid the only fermentation product of the homofermentative Lactobacillus plantarum? No, it's not. The deepest characterization of our research and development department highlights a lot of other metabolites that play a very important role in controlling the other microorganisms. Through the production of organic acids, diacetyl, hydrogen peroxide, and bactericins, Lactobacillus plantarum is able to impose itself as the dominant bacteria of the silage, fighting all the other unwanted microorganisms. If you think about it, the production of silage, as the production of milk, requires a deep knowledge and management of a living creature. If you want to produce a high quantity of good milk, you need to select the best animal for your batch, and then taking care of them. Emulating the enthusiastic work of a fine farmer, we selected bacteria with an excellent genetic, healthy condition, and high pure fermentation. As we strongly believe in the supremacy of the homofermentative bacteria for the silage fermentation and next to the industry, we focus our efforts in identifying the best strain inside the family of Lactobacillus plantarum. Today, we are glad to have found LACTOSIL, Lactobacillus plantarum 14D CSL. That's the name of the best strain of the best family of lactic acid bacteria for the fermentation performance. Our work was inspired by the geneticists who over the years have been selecting the best cattle for milk production as the Frisian. Trying to emulating them, we started from the best species, Lactobacillus plantarum, so the boss taurus of the bacteria, identifying the Frisian of the bacteria, the LACTOSIL. Year after year, according to the new discoveries of our scientists, we modified the formulation of LACTOSIL with different generations of the product. We started from a blend of different Lactobacillus plantarum strains isolated from forage and silage with the first generation of LACTOSIL. Then we isolated the best 4 strains with LACTOSIL 2. Today, LACTOSIL 3.0 is the unique comprehensive product with the best Lactobacillus plantarum strain. Today, LACTOSIL is a comprehensive starter culture for all your silage, maize, grass, and leguminous plants. The dominance of our strains, together with the highest dose on the market, makes LACTOSIL an essential product. Moreover, our lactic acid bacteria was isolated from plants, and so it's organic farming friendly. If you're looking for a practical and essential way to improve the nutritional value of the silage, without bad surprise at the opening of the silo, LACTOSIL is what you need. During this presentation, I made a short summary of the most relevant factors for the silage management. I hope to have the chance to meet you all and go in depth about the single topics for which you have more interest. I invite you to submit the questions right now, or alternatively, you can think about it and send me the questions to my personal contact. Share with you all the contacts where you can send your questions, and thank you so much for your attention and your time.